Antibiotics have transformed the landscape of modern medicine, but lurking in the shadows is a formidable adversary: bacterial resistance. While penicillins and cephalosporins are the familiar champions of the Beta-Lactam family, today we shine a light on the unsung heroes, other Beta-Lactams crafted to outsmart resistance and safeguard lives. At the heart of all Beta-Lactams lies a critical structure, the Beta-Lactam ring. This ring is a game-changer, irreversibly inhibiting bacterial cell wall synthesis by targeting penicillin-binding proteins (PBPs). However, bacteria have evolved, deploying beta-lactamases enzymes that cleave this vital ring, rendering our antibiotics ineffective. In response to this escalating threat, the pharmaceutical world has introduced innovative solutions known as other Beta-Lactams. Let's explore three key players. Beta-lactamase inhibitors, for example, clavulanic acid. These are the suicide substrates. They sacrifice themselves to protect their partner antibiotics, allowing them to work effectively. Carbapenems, for example, imipenem, often referred to as the big guns. These agents are resistant to most beta-lactamases and boast the broadest spectrum of activity. Monobactams, for example, as trinum, these precision snipers target only gram-negative bacteria, sparing the beneficial gut flora. These drugs serve as last-line agents for treating multi-drug-resistant infections, particularly in high-stakes environments like ICUs, burn units, and among cystic fibrosis patients. However, misuse can accelerate resistance, making it crucial to understand their mechanisms in clinical applications. Consider a septic patient who is failing on ampicillin. What's your next move? Stay tuned as we unravel the options. Bacteria are cunning adversaries in the ongoing battle against infectious diseases. As we proceed, beta-lactamases function as the bacteria scissors. They hydrolyze the beta-lactam ring, rendering essential drugs like ampicillin ineffective. This enzymatic resistance is particularly concerning because it undermines the very foundation of antibiotic therapy, which relies on the ability of these drugs to inhibit bacterial cell wall synthesis. In response to this challenge, early strategies involve combining beta-lactamase-resistant penicillins, such as amoxicillin, with broad-spectrum agents like ampicillin. However, these approaches ultimately flop for several reasons. For instance, combinations of methicillin and ampicillin have historically failed because resistant penicillins are unable to penetrate the protective outer membranes of gram-negative bacteria, which act as formidable barriers against antibiotic action. Poor penetration into gram-negative bacteria. Their thick outer membranes create a formidable barrier that prevents effective drug delivery. These combinations often required sky-high doses to compete with the overwhelming activity of beta-lactamases, leading to increased toxicity and side effects. Alarmingly, some of these drugs could even induce the production of more beta-lactamases, exacerbating the very problem they were designed to combat. Enter clavulanic acid, a remarkable Trojan horse derived from the bacterium Streptomyces clavularis. While it is a weak antibiotic on its own, it serves as a potent beta-lactamase inhibitor. At its core, clavulanic acid features a beta-lactam ring, a four-membered structure that's highly strained, making it incredibly reactive. This strain is what allows it to effectively inhibit beta-lactamases, the enzymes that bacteria use to resist antibiotics. Fused to this beta-lactam ring is the oxazolidine ring, a five-membered structure that enhances clavulanic acid stability and effectiveness. This unique combination is key to its role as a beta-lactamase inhibitor. And like penicillins, clavulanic acid lacks a thiazolidine ring. This absence is significant. It alters the way clavulanic acid interacts with bacterial enzymes, allowing it to serve as a strategic ally in combination therapies. In essence, clavulanic acid is a clever design in the battle against antibiotic resistance, using its unique structure to outsmart bacterial defenses. A true hero in the world of medicinal chemistry. What about exploring the hydroxyethyl side chain of clavulanic acid and how it plays a critical role in its function as a beta-lactamase inhibitor? Let's dive into the structure-activity relationship, or SAR. This bulky hydroxyethyl group prevents beta-lactamases from efficiently hydrolyzing the acyl enzyme intermediate. This forces the enzyme into an irreversible inhibition pathway, also known as suicide inhibition. The OH group forms hydrogen bonds with active site residues, like serine 130 and lysine 73 in beta-lactamases. These interactions help to lock the enzyme in an inactive state, preventing it from regenerating. The 2R5R configuration is essential for activity. If we invert SC22 to S, we lose inhibitory activity. And if we remove the OH group, that leads to a complete loss of irreversible binding. And now we're diving into the mechanism of action of clavulanic acid, a key player in the fight against antibiotic resistance. Let's explore how its unique chemical structure enables it to inhibit beta-lactamases. Step 1. Recognition and binding. The beta-lactamase enzyme, like TEM1 or SHV, mistakenly recognizes clavulanate as a substrate. The enzyme's active site serine 70 nucleophilically attacks the carbonyl carbon C double bond O in the beta-lactam ring. Step 2. Acyl enzyme intermediate formation. The beta-lactam ring opens, forming a covalent acyl enzyme complex. And like penicillins, clavulanate strain structure prevents hydrolysis, keeping the enzyme inactive. Step 3. Irreversible inactivation, suicide inhibition. A clasin rearrangement occurs, forming a resonance-stabilized enamine. The enzyme's active site is permanently cross-linked, meaning it cannot regenerate. The result: one clavulanate molecule destroys one beta-lactamase permanently. So, why does this matter clinically? Synergy with amoxicillin. Clavulanate sacrifices itself to protect amoxicillin from degradation, enabling augmentin to effectively kill beta-lactamase-producing bacteria like S. aureus and E. coli. Meet sulbactam, the ingenious creation where medicinal chemists took penicillin's foundational blueprints and engineered a beta-lactamase killing powerhouse. The sulfone's double-bonded oxygens provide a staggering 300% increase in electron withdrawal compared to the thioether in penicillin. This hyperactivation of the beta-lactam ring lowers its pKa to 5.2 compared to penicillin's 8.3, making it significantly more reactive. A lower pKa means the molecule is more likely to donate a proton, enhancing its reactivity and making it a more attractive target for beta-lactamases. The bulky SO group forces beta-lactamase enzymes into a strain conformation, creating an irreversible molecular bear trap. This results in a half-life of inhibition of just 0.8 minutes, effectively locking the enzyme in place. Sulbactam's design isn't just about chemistry, it's about clinical effectiveness. The sulfone structure stabilizes sulbactam against chromosomal beta-lactamases, ensuring it remains effective in various environments. However, it is deliberately unstable against plasmid-mediated enzymes, allowing for targeted action against specific bacterial threats. Chromosomal beta-lactamases are often more stable and less prone to mutation, while plasmid-mediated enzymes can evolve rapidly, making them a significant challenge in antibiotic resistance. In a 1:2 ratio with ampicillin, sulbactam exploits synergistic pharmacokinetics, with a half-life of 1 hour for both drugs. This combination penetrates areas where clavulanate struggles, effectively targeting resistant strains like Acinetobacter and Bacteroides. Clinical data shows 87% efficacy against beta-lactamase-producing Staphylococcus aureus in skin infections. Despite its strengths, sulbactam has limitations that prevent it from being a standalone solution. Gram-negative bacteria have evolved gatekeeping mechanisms, excluding sulbactam from entering through the Oberd porin. Introducing tazobactam, the evolution of sulbactam, engineered to take down even the most formidable beta-lactamases. Triazol's nitrogen atoms boost electron withdrawing capacity, further destabilizing the beta-lactam ring. This leads to an even lower pKa, making it more reactive against beta-lactamases. The bulky triazole ring creates additional steric strain, enhancing the molecular bear trap effect. This forces beta-lactamases into a strain conformation, making them more susceptible to irreversible inhibition. The bulky triazole ring creates additional steric strain, enhancing the molecular bear trap effect. This forces beta-lactamases into a strain conformation, making them more susceptible to irreversible inhibition. Tazobactam effectively targets a wider range of beta-lactamases, including those from resistant strains like Pseudomonas aeruginosa. Its design allows it to penetrate bacterial defenses that sulbactam struggles against, making it a more potent ally in the fight against resistant infections. The 1:4 ratio with piperacillin maximizes pharmacokinetic synergy, enhancing overall efficacy. Together, they create a formidable duo against serious infections, including complicated skin and soft tissue infections. Some strains of gram-negative bacteria have mutated porins that limit tazobactam entry, a challenge that sulbactam also faces but is exacerbated with tazobactam's larger structure. Bacteria are evolving new beta-lactamases that can hydrolyze tazobactam, necessitating ongoing research and development to stay ahead of resistance. But the battle against resistance is far from over. What's next in the pipeline? Stay tuned for the next episodes of other Beta-Lactam section part 2, and now we have question section.